Good morning, everybody. Great to be here. What I'm trying to do in the next 15 minutes is tell you a little bit about Spirovant and what we do. I'm the Chief Scientific Officer of Spirovant. We are a company, we are a gene therapy company, trying to really develop new medicines and revolutionizing treatments for respiratory indications with a genetic functional underpinning. So on this slide gives you a snapshot of again, who we are and what we do. Our lead program is SP101. That's an AAV vector for the treatment of cystic fibrosis. Cystic fibrosis is also the lead indication we are working on these days. We have a very experienced management team I'll talk to in the next slide in a little bit more detail. We are a subsidiary of Sumitomo Pharma, a Japanese pharmaceutical company. And we're about 40 FTEs spanning different functions in the organization, including research and non-clinical, CMC, clinical, quality, and regulatory. So I mentioned our management team. So we have a lot of experience in our management team in large pharmaceutical companies, but also smaller biotechnology companies in larger disease indications, in rare diseases. And again, the experience also spans across different modalities, large molecules, small molecules, certainly also cell and gene therapy. And I also want to mention that we are blessed by really the support of a very experienced and accomplished founder team and scientific advisor team, which is largely located at the University of Iowa. That's also the team that invented most of the technology we are now taking forward in our SP101 cystic fibrosis program. People like Michael Welch, who just received the Shaw Prize for Life Sciences and Medicines for his really groundbreaking investigations and discoveries in cystic fibrosis. People like, again, Joe Sabner, who was the first principal investigator of the first gene therapy study in cystic fibrosis, which again, was very well tolerated. It was an AAV study, but again, did not have the expected efficacy. And so based on the combined experience of that team, we are really taking forward what we believe is a highly differentiated next generation technology for the treatment of cystic fibrosis. And I'll talk about this in a moment. This is our pipeline. Our lead program, as I mentioned, is SP101. It uses an optimized AAV vector, and we are delivering the CFTR gene into the airways of patients with cystic fibrosis. Maybe just a few words about cystic fibrosis for those who are not so familiar with the disease. Cystic fibrosis has the main phenotype is in the airways, and it's represented by really thick mucus in the airways, which can't be taken out of the airways. So you get a lot of chronic infections, you have chronic inflammation, lung function declines very rapidly in these individuals. You have acute exacerbations, acute episodes, which are very often triggered by viral infections. And there are small molecule modulators in that space that treat most of these mutations, but there are about 10% of the patients who do not respond to these small molecule modulators. And that's the primary target population we are going after. These are about 8,000 individuals worldwide, so there is a significant need still in cystic fibrosis. We also have lentiviral technology in the organization, which we are also applying to cystic fibrosis. Now, this is an integrating vector, and the concept here would be that a one-time administration would lead to a lifelong cure of cystic fibrosis. We are also leveraging our expertise and technology to look at other diseases like primary ciliary dyskinesia. Now again, this is a disease of the airways that is also associated with mucus accumulation, very similar to cystic fibrosis. In that case, though, the ciliary motor doesn't work, and so the cilia in the airways they can't transport the mucus out of the airways and clear the airways. So very similar phenotype as in cystic fibrosis. And then surfactant protein disorders is a really deleterious disease in babies, which come down with severe respiratory distress. There's a very high mortality and a very high, I think, clinical need in that indication. And on the slide, it shouldn't read AAV. We are really applying lentiviral technology to address surfactant protein disorders. But let me tell you a little bit more about our lead program, SP101, on this slide. And let me start with just explaining the key components of what we are trying to take forward here, which we believe is quite differentiating in comparison to what others are trying to do in that space. So we have a new AAV capsid, 2.5T. It's a new recombinant capsid that has been selected by directed evolution on cultured human airway epithelial cells. And that's really important to mention because we are really trying with our approach is being as close to human disease as we can be. So this translational aspect is critical. And the capsid we have is very good at transducing human airway epithelial cells from the apical side, because what we are trying to do is give the vector via inhalation, so it has to go into the airways and get through the barrier function of the epithelium. And we believe we have a capsid which can do that. We also added a strong synthetic promoter that is driving robust transgene expression in the airways, in human airway epithelial cells. We packaged a human CFTR mini gene because of the packaging limitations of AAV. A full-length gene takes up too much space in the vector. So this mini gene maintains the full biological activity of the full-length CFTR gene. I should mention CFTR is an ion channel. It's a large gene, and its function is really to hydrate the mucus in the airways. And what I think is very special about our program is that we're also co-developing a small molecule augmenter. It's doxorubicin. And we believe this is critical to really drive the efficacy of our gene therapy approach. And this is exemplified in the cartoon on that slide. So the plan is in the clinic to deliver SP101, the vector, via inhalation into the airways, and then follow up with a second administration of doxorubicin, a small molecule, also via inhalation at a fairly low dose. But we have shown we have a lot of data, and this is exemplified on the next cartoon, where we can show that while our vector has great tropism for airway epithelial cells, it needs another kick to really translocate from the cytoplasm of the cells into the nucleus to really drive a very efficient transgene expression and CFTR function. So this is where the small molecule augmenter is coming in. It's an intracellular translocation step triggering the translocation of the vector into the nucleus. Now, on the next 3 slides, I want to show you a little bit of preclinical data exemplifying what I'm talking about here. So these are these human airway epithelial cultures I mentioned already. So you can take cells from the airways of individuals with CF or normal individuals, culture them, and in culture they really adopt an architecture which is very reminiscent of the airways. So there is a luminal side which is air exposed, and then a basolateral side. And this is really a workhorse model we are using and others have used in the field, like Vertex for the small molecule modulator discovery. So we use that model. We can use, again, cells from individuals with CF with different mutations in the CFTR gene, put it in culture. We give the vector from the apical side, which is the luminal side of the airways. We add the small molecule augmenter, and then we measure CFTR activity and function. And this is what you can see on the right side here. You can measure this because it's an ion channel by the current that is produced across the barrier function. And you can see that there is a very nice dose-dependent increase in CFTR activity to the level you see in normal airway cultures, which are the controls on the right-hand side. And you can also see how important the co-administration of the augmenter is in order to drive expression and to get activity in these cultures. Now, we have also done studies in a preclinical in vivo model. The model we are using is quite unusual. We are using the ferret. And there is a CF ferret model that has been developed also by our founders in Iowa. They also developed the pig model, but we use ferret because our capsid is tropic for the ferret airways. And I want to show you some of the data we have generated in these ferrets. So we use a very similar approach as we are planning to do in the clinic. We apply the vector via inhalation in ferrets, and then we follow up with a second inhalation of the augmenter. And what you can see here is transgene mRNA expression in the airways of ferrets. You can see that in comparison to no treatment, which is the very left side there. In the middle, these are wild-type ferrets. We get very nice transgene expression. Actually, we reach levels of endogenous ferret CFTR, which is really encouraging to know. And I think, again, a great sign that we would be able to achieve levels that drive efficacy in human. And on the right-hand side, you see that this expression is also durable. So the latest time point we looked at is 90 days or 12 weeks. And we see quite durable expression in the airways, which is also an important prerequisite for a successful efficacy in the clinic. Now, one important question is, and I told you about the mucus accumulation in the airways, is our vector able to penetrate through this mucus layer? Because if not, then you never get to the cells, you never get transduction, and you would never get any form of efficacy. So in this ferret, there is a CF ferret model that is actually has very similar, I would say, pathophysiological characteristics like human CF, and that includes the accumulation of mucus. So in a few CF ferrets, we have done exactly the same study here, and this is shown on the next slide. In dark blue, this is administration of the vector in CF ferrets, which already had accumulated mucus in their airways, versus non-CF or wild-type ferrets on the right-hand side. And you can see that we get a very similar efficacy as measured by mRNA expression in the CF ferrets, demonstrating that, again, we believe the mucus is not a barrier for our gene therapy vector, but we can dose through it. So in summary, we are trying to really take all and leverage all our technology experience and our expertise in the company, which is spans, I think, the 4 areas here shown on that slide, which is technology and assets. We have a lot of experience in vector engineering, transgene optimization, and proprietary vector design with regulatory elements. We have a lot of experience in the organization in lung biology and translational medicine, and I want to highlight the translational medicine aspect. We are trying to get as close as we can to the human condition, as is exemplified by the human airway epithelial cell culture data I've shown you. We are big believers in translating from the clinic back to the bench, so I think getting good biomarker data when we are in the clinic and we're just getting ready for the clinical studies. We just achieved IND clearance for our lead program, actually. And so we are really getting ready for it and can also engage into a great network of KOLs and patient advocacy groups. And in particular, this is the Cystic Fibrosis Foundation, which is a great support group for our program. And we have experienced in CMC, in analytical methods. We can produce batches up to support GLP toxicology studies and have great relationships to CDMOs. And device is very important in our case. We have experience in nebulization devices and the formulation, and this is what we are also now leveraging for other indications like surfactant protein deficiencies and primary ciliary dyskinesias. And with that, I'm going to close. I see I have 15 seconds left, so I made it. Thank you very much for your attention. I'm happy to take any questions, and we are open for business. Thank you so much.